Next up is oral presentation 24, San Antonio, Texas, Team 2. It's a Mission 7 flight experiment with 12th grade student researchers.
Hello, I'm Caitlin Block, and this is John Gonzalez. We're representing Business Careers High School down in San Antonio. We chose to study the rate of cell growth and division in plants in microgravity conditions.
How does microgravity affect the rate of cell division in plant cells?
So we chose to study specifically cell division in radish plants. We chose them because they have a relatively short time that they need to germinate in comparison to other plants. What we want to see is the differences in the size and number of cells, and we want to see if there are any irregularities in the cells that are grown in microgravity. We're hoping that this can provide insight into whether or not it's feasible to produce enough food for long-distance space flights in the future. So this would give us an idea of how many plants could be grown in what time period. To prepare for experiment design, we studied gravitropism, which is the movement and growth of plants in response to gravity. What we found out, we learned about the starch-statolith hypothesis, which states that there are specialized amyloplasts within the root cap cells of plants. They're called statoliths. Gravity pulls them down to the end of the plant cell, and that triggers the release of the plant growth hormone auxin that tells the plant which way to send its roots and which way to grow the stem. That's how plants sense gravity. So we know that gravity triggers that growth in plants, and we want to see, without gravity to trigger that, if plants will still grow at the same rate or if they'll grow at all.
Some of the variables that we have that go along with this is that considering that there's the control variables that we have is there's no light in the space station, and the temperature that we have for the seeds is another variable that we have, and the amount of water that it gets and the gibberellic acid that we have to stop the experiment so when it comes back during its return flight, it doesn't affect the seeds. The independent variable in our experiment is gravity. As Caitlin said, we wanted to see how it would affect the seeds without gravity. And the dependent variable is our rate of growth. So our hypothesis is, without the force of gravity, will plant cells divide at a slower rate, but will not stop dividing completely?
So we had a type 3 FME, meaning that it has 3 chambers separated by 2 clamps. In the first one, we had the radish seeds along with cotton to wick the moisture so that they don't get too much all at once and drown. Our second volume contained deionized water and also gibberellic acid, which is a plant growth hormone, to hopefully nourish them so that they're able to germinate quicker and also grow more. Our third volume contained formalin, a biological fixative, to kill and preserve the seedlings so that they're not affected by Earth's gravity during the return trip.
Some of the procedures we had was for prepping the FME tubes to begin with. The second step in this is opening the first clamp to allow the gibberellic acid to mix with the seeds. The third step is opening the second clamp so that the fixative can stop the growth after our experiment has been finished. We would have gotten to step 4, but unfortunately, we didn't make it that far. So we wanted to acknowledge some of the sponsors we had and everyone else that have helped us along the way, especially the SSEP and the Smithsonian National Air and Space Museum for having us here. Thank you.